What exactly is the nervous system? The nervous system is the network of nerve cells and fibers that transmits nerve impulses between parts of the body. Your neurons play an important role in your everyday life.
If you accidentally touch a hot candle, the neurons of your hand, part of the peripheral nervous system, transmit that signal to your central nervous system, which includes the brain and spinal cord. Your brain then sends the signal back to your peripheral nervous system, which results in you moving your hand away.
Neurons receive and transmit information. Here you can see a presynaptic cell and a postsynaptic cell. The cell body contains all the organelles of the neuron. Neurons also have branched extensions called dendrites, which receive signals from other neurons. There's also a single axon which transmits signals to other cells. The base of the axon is the axon hillock, and this is where signals are generated. The branched end of that axon uses a junction called a synapse to transmit information to other cells. The part of the axon branch that makes this junction is the synaptic terminal. Chemicals called neurotransmitters pass the information from the transmitting neuron to the receiving one. The presynaptic cell is the transmitting cell, while the postsynaptic cell is the receiving cell. The arrows show the overall pathway of the information.
The green diamonds are potassium and the red dots are sodium. Each cell has a voltage gradient on either side of the plasma membrane. This difference in electrical charge is called the membrane potential. The membrane potential of a currently inactive neuron is called the resting potential, and this is between -60 and -80 millivolts. Extracellular fluid has a higher sodium concentration of 150 millimolar and a lower potassium concentration of 5 millimolar. In the cytosol, there is a lower sodium concentration of 15 millimolar and a higher potassium concentration of 150 millimolar. These concentration gradients are maintained by the sodium-potassium pumps in the plasma membrane. This process involves ion channels that have selective permeability, only allowing certain ions to pass. A resting neuron has many potassium channels open, but not as many sodium channels open, and thus many more potassium ions leave the cell, leading to a negative charge inside the cell. This negative charge is the source of the membrane potential.
During this time, sodium and potassium channels are closed. Step 2 is depolarization. During this time, a stimulus will open some of the sodium channels. The inflow of sodium through these channels depolarizes the membrane. If depolarization reaches the threshold, it will trigger an action potential. Depolarization opens up most of the sodium channels, while potassium channels remain closed. The sodium inflow makes the inside of the membrane more positive. 4 is the falling phase of action potential. Most sodium channels become inactivated, blocking sodium inflow. Most potassium channels open, permitting potassium outflow, which makes the inside of the cell negative again. Step 5 is the undershoot. The sodium channels close, but some potassium channels are still open. As these channels become unblocked, the membrane returns to its resting state. Potential moves across the axon pictured here. This is called saltatory conduction. In stage 1, an action potential is generated as sodium flows inward across the membrane at one location. In stage 2, the depolarization of the action potential spreads to the neighboring region of the membrane, reinitiating the action potential there. To the left of this region, the membrane is repolarizing as potassium flows outward. In stage 3, the depolarization-repolarization process is repeated in the next region of the membrane. In this way, local currents of ions across the plasma membrane cause the action potential to be propagated along the length of the axon. Type of glia cells, which are cells that support the neuron as pictured here. Myelin sheets are produced by glia cells like Schwann cells. They cover the axon and increase the speed of transmission of the action potential because the action potential can only occur at the nodes of Ranvier. Schwann cells increase the rate of signal transmission by increasing the diameter of the axon and allowing the action potential to essentially jump the nodes of Ranvier.
The following is an illustration of a chemical synapse. When an action potential depolarizes the plasma membrane of the synaptic terminal, it opens voltage-gated calcium channels in the membrane, creating an influx of calcium cations. The elevated concentration of calcium cations in the terminal causes synaptic vesicles to fuse with the presynaptic membrane. The vesicles release neurotransmitter into the synaptic cleft. The neurotransmitter binds to the receptor portion of ligand-gated ion channels in the postsynaptic membrane, opening the channels. When the neurotransmitter is released from the receptors, the channels close.